And recording should be in progress. So, hello, my name is Laurel. I am the Education and Outreach Program Manager at the Finger Lakes Partnership for Regional Invasive Species Management, otherwise known as the Finger Lakes PRISM. We are located at the Finger Lakes Institute at Hobart and William Smith Colleges, which is where I am right now. And thank you for joining the 2026 Macrophyte Survey Program virtual training. So this is the only virtual training that we're going to be doing for the Macrophyte Survey Program. But like I just previously mentioned, you're going to be able to access this recording on our YouTube anytime you want after I have uploaded it. And we're going to have many in-person workshops across the region if you would like to join in on those. But without further ado, I'm going to go ahead and share my slides. We do have a good bit of stuff to go through. So some of these things I might go a little quicker just to make sure we can get everything and we're all good on that front. But if you need me to slow down, just put it in the chat and I will be happy to slow down. I know I can sometimes get ahead of myself and talk a little fast. So I want to make sure everything is good. Alrighty. I'm going to actually move this over here so I can see it better. And you all are able to see my screen. Yes, you can just like do a thumbs up or something. Screen is all good. Okay, awesome, great. We are going to get started. So the Macrophyte Survey Program runs from June 1st to September 30th. So it actually just started this Monday and kicked off the week. So happy you all are here and eager to get started on the Macrophyte Survey Program. It is one of our most popular citizen science programs, so really happy to be doing it this year. Like I mentioned previously, we are located at the Finger Lakes Institute at Hobart and William Smith Colleges, and the goal of the Finger Lakes Institute is to translate knowledge and research about the Finger Lakes watershed into collaborative action. So the Finger Lakes Institute typically has 4 different program areas. We do contaminants, nutrients, watershed management, and where I personally fall within the invasive species component of the FLI. And speaking of invasive species, like I had said, I work for the Finger Lakes PRISM. So we are a network of state, federal, local agencies, NGOs, and other stakeholders, and we support a diverse and comprehensive approach to invasive species management. So we allow partners to share and leverage the limited resources that there are for, environmental things, invasive species in general. And we're funded through the New York State Environmental Protection Fund, which is administered through the DEC of New York State. Our mission is to reduce the introduction, spread, and impact of invasive species within the Finger Lakes PRISM region through coordinated education, detection, prevention, and control measures, which you all are going to be actively contributing to by participating in one of our citizen science programs, or maybe multiple. I welcome you to participate in all of them that you want to. It could be like a Pokemon series, collect all citizen science programs or something. But I feel like for this crowd, I really don't need to go too much into what is an invasive species because if you're here, you probably have some idea. But just to say it, an invasive species is one that is non-native to the ecosystem and whose introduction causes or is likely to cause harm to the economy, environment, or human health. I always really like to show the invasion curve, and hopefully this is a little bit of a blurred image, so I do apologize about that. But generally at the bottom, so that kind of like light blue, that is prevention. So that is mostly like the invasive species aren't here and we don't want them to be here. So that's where we ideally want to be. Then it goes into eradication where they might be here, but we can do something about it and we can take care of that small population pretty quickly. Then goes to containment, which is the next one. It's kind of a peachy color, I would say. But containment is where, okay, they've kind of started spreading more rapidly. We probably can't eradicate them, but maybe we can contain this population. We're not going to let it spread any further outside the state, et cetera, et cetera. But then you get into asset-based protection and long-term management, which is that kind of minty green at the top. That's where it's the species like Tree of Heaven, where they're pretty much here to stay, and we are just looking at long-term management of those species. So we always, of course, want to fall within that first little blue to grayish line of prevention and eradication. That's where we want to be. And by doing this citizen science program, you are actively helping us to do early detection and rapid response of a lot of invasive species that could cause some issues. Go to the next slide. There we go. Okay, so why are we even doing this? Why are we surveying local water bodies? What's the gist of that? Water bodies are high traffic areas. I'm sure a lot of you are very aware that boats are the main form of spread for aquatic invasive species. I also, as part of my job, I run the Watercraft Inspection Steward Program. So they're actively out at the launches, trying to prevent spread, detecting those aquatic invasives, educating boaters about it. But water bodies are high traffic areas, and there's a lot of boats going in and out, and that is going to spread aquatic invasive species and just spread other things that could also be carrying those aquatic invasives. So it's really important for us to understand the communities and species distribution, which I'm slightly jumping ahead of myself, but I'll go ahead and go to number 3. We want to create spatial data to better understand macrophyte communities and species distribution. So by doing the Macrophyte Survey Program and consistently participating in maybe like a same location or in a couple different spots along a water body, wherever you want to go, by you doing that, you're helping us understand that community that is in your lake, in your area, so that we can see the changes throughout the year, but also maybe you're going to early detection, rapid response. Maybe you're going to find something that wasn't previously noted in the area and needs to be addressed. So if you do find something that isn't known to be in that water body, we're going to be able to flag it and get our team out there and see what's going on. So this is also something that for our team, we have 5 full-time staff members. We do have a lot of seasonal staff, but with only 5 full-time staff members and an area that we cover, which is the size of New Jersey, having volunteers like you and citizen scientists are really helpful because that helps us to get a better idea of what's going on in our region whenever we physically just can't get out ourselves. So you guys are part of the team and you're helping us a ton, and we appreciate it a lot. So, how does the Macrophyte Survey Program actually work? How do you do it? Anybody with a smart device and a rake can participate. I will get to rakes in a second. But as long as you basically have a phone or a tablet or some sort of like device that you can use, like a computer, you can do the
I'll
macrophyte survey.
shower quickly after this.
Oops, Julie, I
But
don't think you're muted.
oh my gosh, I'm sorry.
It's okay. But yeah, so anybody with a smart device or a rake can participate. If you need assistance with either of those, you can message me. I'm more than happy to help. And where does it take place? We are going to have workshops across the region. However, this is also an independently run program. So in addition to you being able to come to some of our workshops if you would like to, you can do this anytime you want, anywhere you want. You can toss a rake off of your dock if you have a dock, if you live by the lake every morning. If you frequent the boat launch, you can toss the launches. You can really do it wherever you want to. Consistency is helpful. So if you want to pick a spot where you know you're going to be tossing the rake a lot and doing that repeatedly, that helps us kind of see that distribution. But you can really do it wherever you want, and I encourage you to come to our workshops as well, and you'll meet up with some people that have a similar mindset, really want to help protect the water bodies, and are there for the same reason as you, so they're really fun to do. And how do we collect the data? So the reason that we use a smart device is because we upload it all onto ArcGIS, which is an online mapping service. And it basically just allows us to visualize the data and, of course, analyze it. So by you participating in this and using an online app to record your data, it helps us more to it just makes it a lot easier on us than doing, paper copies and collecting those from 17 counties across the region. So by using your smart device, it's really helpful for us. But you are asked to survey for both native and invasive macrophytes. So understanding the native and invasive communities are important, especially when it comes to the aquatic side of things, because knowing what is there and maybe we start to see these invasive macrophytes coming in and choking out these native communities. So by gathering both of those sets of data, we also know, the biodiversity of the space, if there's potential for invasives to move in, potential restoration opportunities, et cetera, et cetera. It's just helpful for us to know both. So you get to learn two things. And you also get to find a body of water, like I said, and you're going to just toss the rake into the water. Try not to toss yourselves into the water. You toss it into the water. You should have a rope at the end of it. So you're pulling it back in. And you, ideally, probably at this point, maybe not a couple weeks ago, my stewards were tossing rakes and they were pulling up dirt and mud and muck. But now there are probably some plants growing. So whenever you pull the rake back, you're going to most likely have collected some macrophytes. You'll ID them and record them within the Survey 123 form. I do want to acknowledge I'm going to be kind of rapid firing a lot of different species at you during this. I do not expect you to memorize all of them right now. Like I said, this presentation is going to be recorded and you'll be able to view it whenever you want to on our YouTube, but I'm also going to be putting these slides on our website so you can have the reference photos and everything. There's even a longer version of this that has additional ID on all of these species in it that I can share. But don't get too overwhelmed or scared whenever I'm about to show you all of these species, because I promise you're not going to know all of them at the beginning, but that's what's fun about this. You get to learn new things and identify a lot of different stuff. So, if you have been kind of wondering, okay, well, you've been saying rake a lot. Oops, pause for a minute. So if you have been like, oh man, Laurel, you're saying rake a ton. I don't have a rake. I'm not sure of what a rake is. This is going to be showing you how to use a rake. It's about a 4-minute video, so I'll mute myself and turn off my camera for the duration of it, but just watch it. Before we get started, I will say if you do not have a rake and you would like to get one, we are doing a build your own rake workshop in the Finger Lakes region on June 10th. It's at the Interlochen Trailhead in the Finger Lakes National Forest. There's limited spots available, but we will have supplies for individuals to build their own rake. So if you want to get on that list, email me ASAP before the spots fill up. But that's just a simple little plug before we do this, just in case you're struggling on how to get a rake. If you are not able to attend that workshop and still don't have a rake, A-okay, I will just send you the materials that you need, and you unfortunately will have to purchase them, or you can jury rig a rake yourself. You could even, pull up some plants with your hands. Really, you can MacGyver this however you want to, but I'm more than willing to help you figure out how to obtain a rake and how to make a rake. So just send me an email and we'll get it figured out. But I'm gonna go ahead and let this video play, and then I'll see you guys in 4 minutes.
Laurel, if there is supposed to be sound with this, we're not hearing it, or I'm not.
Oh no, there is supposed to be sound. I am hearing it on my end, but
And
potentially did
somebody
not get
said they aren't
translated.
seeing it.
I only
How
saw it
many?
once I turned over to your screen. It wasn't being
Okay.
broadcast to the meeting.
Gosh dang it. Well, the video itself, let me. Okay, so I'll go ahead and just kind of resume, but I think you guys got to see most of this. Michelle put a comment in here about marking the rope with different pieces of tape or marker or whatever. It's really helpful to understand depth, so you can just do those by meters, whatever you want to measure those by, but marking it helps know the depth, and we will be asking you about that. But apologies if the video was not showing for you or if you weren't able to access it. I will admit, I'm an outdoor person, not as much a technology person, so sometimes Zoom still, eludes me on a lot of different things. But if you want to watch this video on your own time, it is on our YouTube. It's an older video, but you just have to scroll back a little bit. It's just really helpful. It shows you how to do a rake toss, just generally what to do for building a rake. But like I said, if you have any questions or want advice on how to build a rake, just send me an email. I am more than happy to help you guys figure out how to do this because I don't want the rake to be a barrier to participating in the Macrophyte Survey Program. So do not think that it is for 1 second. We will get it figured out. And no, stop talking or going. It was talking for me. Every single time I tried to click anything on my computer, it paused and I was like, oh no. But we're going to go ahead before we get into the plant ID. I wanted to just run through the survey 1, 2, 3 stuff really quickly. So I have this pulled up. If you do not already have ArcGIS Survey 1, 2, 3 on your phone or your device that you're going to be using, I recommend you download it. This is what it looks like in the App Store. It's like a green notebook with a check mark, ArcGIS Survey 1, 2, 3. Whenever you're doing this survey, you can click Continue without signing in. We wanted to make sure that this was accessible to everybody, so we're not making you sign in or anything. It's at the bottom, so it has that sign in, manage ArcGIS connections, but then there's a little thing at the bottom that says Continue without signing in. You can click that for all of our surveys. This is the QR code that should allow you to get the Finger Lakes Prism Macrophyte Survey Program onto your device. So all you have to do is scan this QR code and you are good to go. If you are having issues scanning the QR code, I can send the link to you as well. And let me just check the chat really quickly to make sure. Yes, I absolutely will send a link to the video to all of the people here. But yeah, I will go ahead, just give you guys a couple seconds to, if you haven't already, scan this QR code. Make sure you've got it down. After we've done this, I'm going to share my screen that has the survey on it. So we're going to kind of just go through all of the stuff on there and make sure that you all are knowing what you should be doing. So I'm going to go ahead and stop sharing this screen, and then we're going to can send the link. Yes, I can send the link. Let's see if it'll let me send it through here really quickly. Do, 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 do. Copy link. And here is the link. So if you click that, it should download onto your device. If it doesn't work while we're doing this training, don't worry too much about it. We can figure it out at a later time. I want to make sure that we get through the main gist of what the survey is, and then we can troubleshoot later. I'm happy to get on calls with people if you need to. But yeah, I'm gonna go ahead and share this screen. Alrighty, you guys can see my screen too? Great. Okay, so I'm going to be looking over here because it's on another screen for me, so I'm not ignoring you guys, I promise. But we have the sampler name. So at the beginning of every survey, you can do your first and last name. It just helps us if you keep that consistent across all boards. So if you have a nickname that you've been using with us and you've been participating before, continue using that. It just helps for consistency across programs so we can make sure we acknowledge the work you're doing. We do give out prizes and appreciation tokens, so just want to make sure we acknowledge that. Your IMAP Invasives Observer ID. So all of our data is shared with IMAP Invasives. If you have an IMAP account, you can put that in here. If you don't, you do not need to worry about it, but we do share it, so it eventually does get uploaded to IMAP. And if you want your name to be on it or your IMAP Observer ID to be on this data whenever it uploads, then you're going to need to put your ID on here. If you would like to, I'm going to show you guys how to save your favorites super duper quickly, just so you don't have to keep introducing, you don't have to keep filling your name out. So I'm going to put my name in, and this is not my ID, but I'm just doing it for the purposes of this video. You can go up to the corner, these 3 lines right here, and you can click Set as Favorite Answers. So now these answers are going to be saved in your favorites. All you have to do is click the X. You're going to save in your drafts, and then whenever you go to collect another one, all you have to do is click in the top 3 and then paste answers from favorite, and your answers are right there. It's really easy and just saves you time from having to input your name again and again or your IMAP user ID again and again, and it helps across all of our programs. So that's just a helpful little trick that hopefully will help you. After that, your device location services should be on for this app, so just make sure you've got your device location on. It'll automatically take your location. I'm using my computer right now, so the map is not popping up, but as long as you're on your device and your location services are turned on, you should be, it just will automatically do your location. You could also zoom in to locate the sampling location, so you could just zoom and click where you are on the map, but it should just be taking your data as well. You can also enter the coordinates manually if you're having issues with it. It's just another additional way of making sure that, you know, we're getting that accurate detection area, because if you detect something that's high priority and we don't know where it is, then it's really hard for us to go to that space. So just make sure your location is on and you are accurately recording where you are. Water body, you just click down here and you can click whichever water body. I'm going to go ahead and just go to Seneca because I'm currently on Seneca Lake, and that's the name of the water body, or you can fill in using the other. So if you're at a space in the Finger Lakes region, which is not listed on here, which could be a lot of places, you can just click the other and input where you are in the region. Then we go to observation type. So how'd you collect or observe your sample? Donated rake on a rope, handmade rake, other rake type. So this could be, you know, your MacGyver rake. Manual removal from substrate with hands. So that's another method. You can literally just dip your hand in the water and pull it out and see what you get. These are good rakes too. So you could also do it that way if you feel like leaning over the dock. And then visual, so no contact with plants. Maybe you just glanced down and you saw something and you were like, whoa, I know what that is, and you recorded it. That's also good too. But rake is obviously the preferred method. However, these other methods are good and fine to use as well. Observation location. From where did you collect or observe your sample? So this could be from a dock, the shoreline, off of a boat. Maybe you were taking a nice joyride and you came across a bunch of hydrilla and you hated it because it got in your motor and you wanted to let us know about it. You should let us know about it anyway, but that's another reason. So just make sure you let us know where you did it from. So I'll click shoreline for now. Water depth. So this is where those, you know, increments on the rope could be really helpful. So we're asking for feet. If you want to just mark off foot by foot with some sort of tape or some sort of, you know, permanent marker, that's just great for us to know because different plants grow at different depth levels. It's just another helpful bit of information whenever we're going through and analyzing everything to understand the communities better. So water depth and substrate. So what substrate was your sample collected from? Mucky, rocky, sandy, clay, organic debris, or mixed. This is kind of your own judgment. I will note that this survey is used by our field crew as well. So some of these questions, if you're not sure, you know exactly what substrate it is, and you just want to take a guess, that's okay. But again, this is used by our field crew, so there are some more specific questions in here as well. And rake toss data. So we've got macrophyte density. 0, trace, sparse, medium, dense. We do have photos here. So if you click on the photo, it'll show you kind of what, you know, each of these rakes looks like. And you'll be able to just mark how much you think is on the rake. Again, this is an estimate. So it doesn't have to be, you don't have to measure exactly out everything, but just a general estimate of this would be great. You're also going to notice that the substrate is optional where the macrophyte density is not optional. So again, and then let's go to here. Let's do sparse. So the rake was sparse. Total species present. Let's see. Let's just pretend we found American eelgrass, coontail. Oh no, we found some curly-leaf pondweed because that's out everywhere right now. We have that too. So just doing a couple. Oh no, no, oh my gosh, I'm so sorry. Yeah. But you can do it one species at a time by putting it in here. And then percentage of pool. What percent of the total pool was the species? And just, 50%. Let's do 50. And there we go. And then you just add, and then you can do 2 of 2. So here's the next one. We had curly for the first one. You can do coontail. Let's go 25% coontail, and I added that. And then my last one was, what did I want? American eelgrass. Let's do 25% American eelgrass. And it adds it for you, so you don't have to add it yourself. You can just, well, you do have to add it yourself, but it tells you whenever you hit your limit, so you don't have to keep remembering, everything in. But just use a general estimate. So something would look like, 50% of it is curly leaf, and then the rest of it is like a even mix between American eelgrass and coontail. So that's how I would generally estimate this. Again, doesn't have to be the most accurate thing in the world. You should be consistent with your data, but just make sure that you're getting, a good idea for us. Sample collected, this is just where, if you found hydrilla, then we would want to know, and we would want to take a sample so that we could confirm the ID. So if you did collect hydrilla and you wanted to give us a sample, you could say yes. You could just do other and then sampler name and then put down what it was. You would just send me an email if you did find something, just take a sample, then that way I know where it was. And you can put the same, just sampler initials, date, sample type, number. So this is just an example of what it could look like. But as always, email me immediately if you find hydrilla. It's just, something that we want to make sure we're on top of if we do find it. But this is where you could take a sample and indicate in the survey that you did take a sample of it. And then photos. Photos are required for all of the surveys, and it's required for this one because we can't confirm an ID if we don't know what it looks like. I 100% believe that you guys all, probably know some aquatic species, and after this you're going to be pros and whizzes at it, but we just also need to confirm it because it does get uploaded to IMAP. We need to confirm it on our end and make sure that all detections are accurate. So you have to take a photo of every species that you see. It just helps us for that. So you could lay out everything and then take a photo of those plants. There will be some more information on taking like what parts of the plant you can take photos of, but this is generally the survey. It looks pretty similar, I believe, to what people have been used to in the past. We wanted to kind of reuse some of the things that we had before because they worked really well, but this is what the Macrophyte survey it looks like this year. I'm going to go ahead and just check the chat super duper quickly to make sure. I'm not sure if I can change the screen type to phone or tablet. I do apologize about that. However, reshow QR code will do. Already have survey 123. What is the name of the survey for the search bar? It is the Finger Lakes Prism Macrophyte Survey. It is, I'll show it to you really quickly, close and loose changes. It is this one, Finger Lakes Prism Macrophyte Survey Program. Owned by Sam, Becca Anderson, HWS, and it should have all of this information up here in the corner. Alrighty, I'm going to stop share of this screen, and then I'm going to go back into my PowerPoint. There we go. Here is the QR code again. Alrighty. And once we are all good on this, I'm going to move into our next QR code before we're going to get into some species ID and everything. So we have one more QR code that I'm asking you to fill out. Again, I can send links to these. I'm going to be doing a big email after this too, so there's going to be a lot of information packed into that for you all. So if you are missing something or if you don't have these surveys immediately right now, I'll be sending an email to all of you guys. So don't worry, we'll get that on your phone. We'll get it uploaded. Again, my email inbox is always open. If I don't respond within a day or two, just poke me again. It might have gotten buried. But this is the how to fill out volunteer hours form. So this is a little different than previous years. We're trying to get all of the different volunteer programs under one blanket of, tracking hours, just so we know how much time you guys are putting into it. So all you have to do is just scan this QR code. It's a very simple survey. I'm not going to share it, but it just asks you to put your name, the program that you are volunteering for, and the amount of time you spent volunteering for that program. It's very, very simple. Just do it after you're done. For this program, you can maybe do a rake toss a day. Maybe you want to do a couple rake tosses a day. This one might not be quite as lengthy as some of our others, like the trail survey program, where people are out actively, they're hiking for however long they want to hike. But for this one, we just are trying to consolidate a lot of those hours. So making sure that everything is good on that.
Lindsay, could you scroll down or up a little bit? The QR code is cut off. There, thank you.
Whoops. Perfect. Is it showing up for the trail survey program? We might have had an issue for this last time. Did somebody...
Yeah, it is. It's the trail survey.
Gosh dang it, not me. I'm trying to get you guys to sign up for the trail survey too. Not actually, but okay. I will send you all the volunteer form. But hey, now you have the trail survey on your phone, so if you want to do that too, you can. But yeah, I'll send you guys the volunteer form. But again, it's a very simple form. It just goes straight on through. I've been using so many QR codes, they get all mixed up. Come on. Okay. Now the fun part of me spitfiring so many different aquatic species at you. These slides were provided by the Aquatic Invasive Species Program Manager, Catherine Farrell. She made these for training. They're really, really awesome and wonderful, alongside Claire McMahon, who is our Aquatic Invasive Species Fieldwork Coordinator. So just shouting out to them on providing me with these slides. But we're going to go ahead and get to some aquatic species ID. Again, do not feel alarmed if you are getting a little overwhelmed. There's a lot going on here, and I'm just going to try to cycle through all of them. You are more than welcome. You're going to be able to look at these anytime you want to after this training, but this is just an opportunity for you to ask questions in the meet, like right now, and also to participate. So, aquatic species identification. Before we get started on, the actual species, I want to talk a little bit about the parts of the plant. So the parts of the plant are really helpful for us to identify what plant you are saying it is. So like the stem, tubers, the leaf or the leaflets, the margin, which is the edge of the leaf. It could be serrated, it could be simple, etc., etc. Turion, which is a vegetative bud capable of growing into a new plant, flowers, and seeds. All of the above are super duper helpful for us when it comes to identifying these aquatic species. So if you can take photos of these identifying characteristics whenever you are, submitting your species, it helps us to be able to confirm your identification. So parts of the plant, that's what that looks like. And then shape of the plant. So they could have alternate leaves, which are leaves spaced singly along the stem. So it's kind of like, if my arm is a leaf, there's a leaf here, and then it alternates to another one here and up here. That's what an alternating leaf looks like the photo. I didn't really need to show you my arm. There is a photo on it. But then there's an opposite leaf pattern where the leaves are opposite each other on the stem. So those are the two that are really common in terrestrial. But then you also have whorl. So there's a whorl leaf pattern, which like you saw in the video, maybe if it played for you, the Elodia is like in a whorled leaf pattern. So the stem is in the middle. There's a central stem, and then the leaves are whorled out around it, kind of like a wheel. So that is what whorled looks like. Then there are rosettes, which are leaves arranged in a radiating pattern from the base of the plant. Water chestnut is a really good, common, one that we think of whenever we think of a rosette because they sit on top of the water and they're right there in that little rosette form. So I feel like I can't not use my hands when I'm talking about these things, but that is what generally the shape of a plant looks like. And the location of the plant. So you guys are going to be tossing a rake. Maybe you're standing on the shore and you're tossing the rake into the water and you get a little further down. So you could get a lot of these different plants on your rake depending on how good of a throwing arm you've got. Maybe you really chucked the rake into the water. So the location of the plant does change what kind of plant you're going to get. So floating has leaves that float on top of the water. There's submersed and submerged, which means it grows underneath of the water. And then there's emergent, which are mostly found in shallow water with a large portion of the stems and leaves growing out the water surface. So there's just multiple different types of aquatic plants that you might encounter while you're out there. And I wanted to just bring that to your attention in this training. Alrighty, we're going to do some high priority aquatic invasive species. I'm sure you guys have heard of a lot of these, but we're just going to run through them really quick. Water chestnut, like I had mentioned, so Trappa natans. Its habitat, it likes ponds, lakes, bays, canals, and slow-moving rivers with muddy bottoms. And it tends to grow in shallow water, and it's rarely found in water deeper than 15 feet. It has that rosette pattern. It has glossy, triangular, toothed leaves with inflated petioles. So, right, can you guys see my mouse? I'm not sure if you can. If you can, then I'm circling that inflated petiole right here. But that's a good identification of water chestnut. They also have those four spine seeds that hurt like a Lego whenever you step on them, probably worse than a Lego. So it has those seeds and they're really, really painful if you do step on them. It also has small four white petaled flowers that can be present in the summer at the center of the rosette. So this is what water chestnut looks like. I'm sure a lot of you have seen it. European frogbit. So this one forms at marshes, edges of ponds, shrub swamps, and wet thickets, and it can form those dense, extensive patches where it completely just takes over the surface. It does really well in calcareous regions. I really hope I said that right. And it is a free floating plant, so it is floating on top of the surface. It has kidney, heart-shaped leaves with purple undersides, and the leaf stems do not have a midline groove like the American frogbit. It has three-petaled white flowers with a yellow center bloom in the summertime. So this is another one that we will be asking you all to look for. Okay, and yellow floating heart. This one lives in slow-moving waters up to 4 meters deep. It can tolerate anaerobic environments and survive in mudflats. So it kind of resembles a water lily. The stems can grow up to 2 meters long, and it has round or heart-shaped floating leaves that grow 3 to 12 centimeters in diameter. The leaves are more of a yellow-green, and they have slightly wavy margins, which you can see over here. They've got those slightly wavy margins about them. There can be 2 to 5 bright yellow, 5-petaled flowers that could bloom anywhere from May to October, so you might see this blooming throughout the entire summer. And it produces a capsule of flat, shiny seeds, which you can see right here. So this is what the yellow floating heart looks like. There's only been one instance of yellow floating heart in the Finger Lakes region, which we had an EDRR response to last year. So if you do see it, it's really important that you let us know about this one. And the American lotus. So this one likes slow-moving water of large lakes. It has one large pale yellow flower that is 4 to 10 inches across on a stalk that rises up to 3 feet above water with a large oval petals surrounding a sponge-like center. So if you've ever seen the lotus pods, they're really, really crazy. But they have large, round, hole without a slit, smooth, and hydrophobic leaves. They can be 12 to 28 inches across, so big. They may rise approximately 1 foot out of the water, and dried receptacles may be seen at the end of the growing season. So you might see this one July through September. And water lettuce. So So this is just a good holistic picture of water lettuce that I wanted to show you before we actually got into everything else. But the habitat, it likes slow-moving water, ponds, lakes, swamps, irrigation canals, or ornamental ponds. It is limited by sensitivity to cold temperatures. Um, its description is a floating plant. It resembles a floating head of lettuce, hence the name water lettuce. The leaf surface is covered by white hairs. It has obvious parallel veins, light green leaves arranged in rosettes, and it's an inconspicuous flower in the center of the rosette. So you can kind of see here, it's a very you can't really see it unless you're super looking close, but it is there. So this is another one that we're asking you all to look out for, which would be an EDRR response. Water hyacinth. Again, here's another just nice, good picture of water hyacinth to fill your screen. Water hyacinth likes slow-moving water, ponds, lakes, swamps, irrigation canals, or ornamental ponds. It also is limited by sensitivity to cold temperatures. It's like a floating plant. It has rosettes of smooth elliptical round leaves. It is bulbous, spongy inflated petioles, so like the water chestnut, but this one is significantly larger. They look like something maybe out of Willy Wonka and the Chocolate Factory. And it also has lavender flower spikes. So this one is another one that we're asking you all to look out for. Here's the big talk of the town, hydrilla. This one is really, we want to know about it if you see it for sure. We're doing some work with the DEC this summer on hydrilla, and it's just something that we're trying to keep from spreading everywhere. It's currently in Cayuga Lake, but it's an invasive aquatic plant of lakes and ponds. It is really prevalent down in Florida, but it's a submerged aquatic plant. It has visibly serrated leaves in whorls of 3 to 8. It's typically 5 leaves in a whorl. It can be mistaken for Elodea. I will get to that later, but it spreads by seeds, tubers, and turions. The tubers are small potato-like structures in the sediment, but hydrilla, if you see it, then it's really important that you let us know about this one. Like I said, it's similar to Elodea, except Elodea does not have the serrated leaves, so hydrilla does. It has those toothed margins, and Elodea does not. Elodea is also typically in whorls of 3, where hydrilla is in whorls of 5. It is rarely in whorls of 3. Alrighty, and starry stonewort. So this one likes depths up to 9 meters in slow-moving aquatic habitats. It is associated with alkaline to neutral pH and has elevated conductivity in North America. It is related to elevated, sorry. It is anchored to the sediment through rhizoids. It can grow up to 2 meters long. It has whorls of 4 to 6 blunt branchlets, and something that I like to do, you can joke that you're killing the starry stonewort because you can pinch it, and whenever you squeeze it, it breaks the cell wall, and it kind of just goes so super limp in your hand. It's really cool if you get starry stonewort and you pop it and it just goes limp. That's a good way for you to tell. Similar to other stoneworts, it can be identified by the presence of tiny star-shaped bulbils or the orange antheridium. I really hope I said that correctly. But you can see it has those little star-shaped baubles for starry stonewort. It can form really dense mats. You can see down here, that's just a ton of starry. And fanwort. So fanwort likes slow-moving streams. It's not widespread throughout New York State. It is a submerged aquatic plant with green, delicate fan-like leaves and occasionally small oval floating leaves. The leaves are oppositely arranged and it has small white flowers. So this is what fanwort looks like. It looks similar to water marigold. Parrot feather! Looks like a parrot feather to whoever named it apparently, but this is a good, photo of what parrot feather looks like taking over a spot. And the habitat of parrot feather is slow-moving waters at depths up to 9 meters. It can survive a wide variety of conditions. It does kind of look like, it kind of looks like there's some parrot feathers, just like really feathered-looking leaves. It is both submerged and emergent leaves, so they can grow down in the water and then they come out. The submerged leaves appear red, orange, and feathery, growing in whorls of 4 to 6. The emergent leaves are stiffer and bright blue-green. It can have small white flowers that may be present along the emergent stem. And then we have Eurasian water milfoil. This one likes ponds, lakes, streams, and rivers. It's widespread and very abundant at some sites. It is in all counties of the Finger Lakes Prism. This one is just it's very widespread, unfortunately. It is a submerged aquatic plant with bluntly tipped and finely divided leaves. The leaves are divided into at least 12 pairs of leaflets in whorls of 4 on red-tinged brown or green stems up to 6 meters in length. It has tiny pink flowers that may be present in June or late July. So that's whenever you would look for those tiny flowers. And then we have brittle naiad. So this one is on quiet water of ponds, streams, and fresh to slightly brackish tidal streams. It is a submerged aquatic plant. It is a submerged aquatic plant. It has a bushy appearance. The leaves are opposite with stiff curled appearance and visible spines along the margins. The seeds are slightly curved and purple with tiny rectangular pits, and it is similar to native naiad species, so there can be some lookalikes for this one, which is again why it's important that you're taking really good ID photos so we can confirm the ID of these plants. This one, curlyleaf pondweed, you guys might already be seeing this already. It starts occurring pretty early in the season of May to July. It likes shallow, alkaline to circumneutral water of ponds, lakes, and streams. It is a submerged aquatic plant that grows up to 5 meters long. It has rigid, oblong, and reddish leaves that grow in alternate arrangement with finely toothed, wavy margins and blunt tips. So this bottom right picture is a really good image of those wavy margins and those like finely toothed edges that it has to it. It has small reddish green flowers on a spike above the water, as well as turions that could be present. So curlyleaf pondweed, it's also like people like to say because its scientific name is Potamogeton crispus, it's like crispy bacon, which sometimes I can see it and sometimes I can't. I waver back and forth on if I agree with the crispy bacon part. But this one is something that you would probably be seeing already. Oh, right. So we're going to be going into native submerged aquatic macrophytes. In the interest of time, because we only have 10 minutes left, I'm just going to be going through these slides and just saying the names of the species. I'm not going to really be giving a backstory of them. However, like I said, these slides and this video are going to be uploaded. So for you to go back and take the time just to refresh your brain on some of these native species is really helpful. There are a lot of pondweeds, and I'm not going to go through every single pondweed. So we're just making sure that we're getting through everything. Checking the chat. Has the FL Prism Field ID book been updated? We are hopefully going to be getting some more of those ID books pretty soon. We don't have them yet, but I do know that an order is going to be in the works for them. So that's a really good point that we will hopefully have some ID books. They'll be available at the office as well as our workshops whenever I do receive them. So if you want to come in and grab one, if you want to come to a workshop and grab one, we'll hopefully be getting those soon. We just don't have them right now. We did run out because they were, as you probably know, very popular, but it's available as a PDF on our website, I will say. So the physical copy, we don't have more of those, but the PDF version of that field ID book is on our website, so you can view that. Alrighty, for the native species, common waterweed, coontail, you guys saw that in the video if you were able to watch it. Whitewater buttercup or crowfoot can be a lookalike for fanwort. Northern water milfoil, lookalike for Eurasian. Slender naiad, all these naiad species can be lookalikes for the brittle naiad. Threadleaf naiad, southern naiad, spiny naiad, which is rare and can be very highly mistaken for the brittle naiad. Cara, muskgrass, or chara, however you want to say it. I have said it multiple ways. This one is one that can be mistaken for starry stonewort, but it stinks whenever you smush this one. It smells like a skunk, hence the name. American eelgrass, water stargrass, small pondweed, leafy pondweed. A lot of the pondweeds look the same. Flat stem pondweed, white stem pondweed, Richardson's pondweed, which can look similar to clasping leaf, which is this one, clasping leaf pondweed, sago pondweed, horn pondweed, northern water plantain, and those are all the native species. I know I completely rapid fired. Again, I apologize. There's just a lot of species to get through. Floating native aquatic macrophytes. And I feel like if I took more than an hour, the training would be a little too long. Mosquito fern, common duckweed, ivy duckweed, greater duckweed, watermeal, variegated yellow pond lily, and then the yellow pond lily, white water lily, and this one should have color. It should say Section 5 lookalike species. For some reason, the color did not translate whenever I did this slide. But like I mentioned, there can be lookalikes for different species. Hydrilla can look like Elodea. So this is a good photo representation of those serrated five-whorled leaves along the hydrilla as opposed to the Elodea, which does not have those serrated edges. The margins are smooth and it typically has leaves in whorls of 3. Turions may be present in hydrilla and Elodea does not have turions. Eurasian water milfoil can look like northern water milfoil and variable leaf milfoil. They hybridize, so sometimes it gets really difficult to determine which one is which because they hybridize and form together. So this is generally a really good way of telling. This one could have 12 to 21 leaflets per leaf. This one has 11 or fewer leaflets per leaf. So this one has a lot more leaflets than the northern. Out of water, the Eurasian hangs very limp and the northern is rigid whenever it's out of the water. So that's another good way of telling it. But these right here, really, really helpful for identification. They've helped me on numerous occasions. Like I mentioned, fanwort has some lookalikes to natives. It can look, I would say, less like coontail. Sometimes it can, but less like coontail. And it looks like water marigold and water buttercup a ton. So these are just some lookalikes for the fanwort that I want you to keep in mind whenever you're identifying it, especially fanwort versus water buttercup. You can see that, you'd have to really look to be able to identify it. It wouldn't be something you could just look at and be like, that's that. You got to get a little closer to it. Then we have brittle naiad, which like I said, can look like spiny naiad, which is a rarer species. So this one, recurved leaves, thin thread-like leaves. It's smaller and has more teeth on the leaves, and it lacks teeth on the inner nodes. And then for the spiny, leaves branch upwards. The leaves are wider. They have large teeth, and the teeth are present on the inner nodes. And I know I just went so quickly through a lot of those species, and it's probably like, I don't even remember half of what you just said. So if you need help with species ID, you can contact me. I am happy to help you. I'm happy to have like a call if you need it. My contact here is lwilliams@hws.edu. You can email me anytime you would like to. You also can reach out to Catherine Farrell, who is our Aquatic Invasive Species Program Manager. Her information is cfarrell@hws.edu. The reference material is on our website under the Citizen Science tab at fingerlakesinvasives.org. I'll be updating that with this slide deck, with this presentation once I upload it to YouTube, which will be sometime tomorrow or Friday, depending on how long it takes for this video to, get through my computer. And then you always could use Seek an iNaturalist. This is a little trickier when it comes to aquatic species. They're not as good at identifying aquatic species as they are terrestrial. But if you want to just mess around with either of these apps, feel free to use Seek an iNaturalist. But if you need help, you always can reach out to us. I'm happy to help. You can reference material on our website under the Citizen Science tab, or you can come to any of the in-person workshops that you would like to. We have, I believe, 7 workshops this year for the Macrophyte Survey Program across the region. The first one is June 15th at the Skaneateles Lake Association office in Skaneateles, New York. But we have a ton of them across the region. I tried to spread them out so there would be spots that people could go to if you want to come. And it doesn't have to be on your lake. It can be a different lake that you come to 100%. You can find all of those dates on our website. I did update our events/calendar tab with all of the dates for the workshops. So please feel free to come to those as well and get a little bit of hands-on experience with that.